All right, welcome to mitosis 2. Here in mitosis 2, we're going to do like a recap of how mitosis works. You're going to practice drawing mitosis again, this time with a different number of chromosomes, just to make sure that you understand it. And then we're going to talk about the how cancer... I totally lost how I'm just going to say this. Anyway, and then we're going to talk about how errors in mitosis can lead to cancer and how we can detect cancers by understanding mitosis and things like that. I hope everybody's staying safe. The CDC has now said that for 8 weeks there shouldn't be anybody around 50 people or more. You know, that includes a lot of movie theaters and malls. I think people are doing a really good job of providing real limited places for that to happen. Remember to keep your distances 6 feet away. And, don't be brave. If you're feeling sick, get better. Stay in bed, drink lots of fluids, and all that kind of stuff. As seriously as we all take this, the less that'll happen. If nothing really bad happens, if it doesn't go into full-fledged, outbreak and stuff like that, we won't see that impact, but don't think that doesn't mean that staying inside was pointless. Had we done the other things, it may have been way, way, way worse. But anyway, let's get to mitosis. So what I want you all to do in your notes is to draw the whole cell cycle from interphase before S phase and interphase after S phase, all the way through cytokinesis, but this time in a cell with an N of 3. So you are going to draw the cell cycle. And that's going to include interphase before S phase and interphase after S phase. Then you're going to draw mitosis, which is prophase, metaphase, anaphase, telophase. And then you're going to draw cytokinesis. Now, I'm going to talk through drawing all of this, but I encourage you to try to do it on your own. I think the hardest part in this one is trying to draw the interphase before and after S phase and prophase. Once you get through the prophase, if you have the right number of chromosomes, then everything else should go fine. It kind of goes in a real logical order. So I would start drawing things and then check up on what I'm doing. And you could also skip to the whole ending and just see how your drawing matches and then, cut it down. So I don't, you decide how you want to do it. You may get to skip ahead a whole bunch in this video so that I don't get people telling me that I'm doing too much with a 30-some minute video, Cody. Hey, man, this is replacing school at this point. Okay, so go ahead and start practicing, and in just a couple seconds, I am going to start. Crack open my Diet Coke here. Okay, here we go. So if this has an N of 3, what that means is I'm going to have 3 unique chromosomes. So I'm going to draw them really short, medium, and then really long. And so because I'm going to have really long ones, I'm going to need to make my cells really big. Okay. And remember that you have one of each. I'm sorry, you have 2 of each because you got a mom and a dad. So 2N is going to equal 6, which means we're also going to have a short red, medium red, long red. Okay. So let's start with interphase. And this is before the S phase when the DNA replicates. That word is important to know. Draw a nice big cell with a nice big nucleus. I've got my short, medium, long. Now remember, it doesn't matter where you draw these things. There's some evidence to say that these chromosomes do sit in very specific positions inside of the cell. Whoops, I drew an extra medium there. Oh boy, this is going to be a problem. All right. I knew it would erase a whole bunch. Darn it. All right, I'm just going to erase that whole nucleus and redraw it. But there's some evidence to suggest that that the chromosomes do sit in very specific places inside of the cells, which is why sometimes you see the exact two chromosomes switching spots with each other all the time. And if the exact two ones are switching spots with each other all the time, then that is evidence that they normally do sit next to each other. So I met one of the scientists that did that research. He works over at MSOE, and it was kind of a mind blower. And, he described us exactly how they did it. It was really fascinating. That guy loves to meet with people and talk about these kinds of things. And I would totally be willing to set up a time that any of you could come and talk to him. He's a really fascinating guy. Okay, anyway, so here's our post-S phase. So we need to have 2 red shorts and 2 red mediums and 2 red longs. And we're going to have 2 black shorts, 2 black mediums, and 2 black longs. I'm going to draw them all right next to each other. Okay, so that is post-S phase. Make our arrows here. Now I'm going to draw prophase. Remember that during prophase, the nucleus breaks apart. So we're going to do that. That we have our centrioles on opposite ends of the cell. All right. 1 centriole, 2 centrioles. Okay, remember that our chromosomes will now pair up. The sister chromatids will pair together to form 1 chromosome. So our 2 shorts will pair together. Our 2 short reds will pair together, and our 2 short blacks will pair together. What do we call that point right there in the middle where they meet? That's right. Way to go. That's a centromere. All right, I'm proud of you guys. What do we call these 2 individual chromosomes that are exactly the same as one another? Ooh, don't do too much there, Liana. Okay, no, those are the sister chromatids. And what do we call, this is a big one here, what do we call these 2 pairs of chromosomes right here where they're the same size and the same shape, they have the same genes in the same places, but they're not exact copies of one another? What do we call those? All right, good job, Imani. Yes, you are correct. Those are homologous chromosomes. Okay? All right. Let's get back here. And we draw 2 mediums paired up together. And then we're going to draw, we don't have a lot of space, so I'm going to draw my 2 longs. 1 here, draw 1 here like this. Okay. All right. So what should be starting to come out of our centrioles? Coming out of our centrioles should be these little fibers, these little strings of microtubules that are called spindle fibers. Okay, forgot to label the name of this. This is prophase. All right, let's move on down to what comes after prophase. All right, good job, Wharton. Yeah, that is metaphase. All right. So we've got our centrioles on opposite poles of the cell. And these microtubules have pushed and prodded these chromosomes around, and until they line up along the equator of our cell. Remember that these don't have to go in any particular order, and that's actually a really important characteristic that'll come into play when we talk about the next part of cell division, which is meiosis. Okay. These spindle fibers should be attached to the chromosomes right at the centromere here. And now we have metaphase. What comes after metaphase? Anybody? Anybody? All right. Yes, Samara, you are right. It is anaphase. And in anaphase, these microtubules start breaking apart. And as they break apart, they shorten. And when they shorten, they separate that pair of sister chromatids into the individual little chromosomes. Remember, it's kind of like yanking you at the belt loop and pulling you backwards. And so that's why I draw them in that little bit of a different shape. Well, that should be our long one on the top there. And we've got another black one. This is our small one right here, then a medium one right here. All right. Now I switch over to my red ones. When you draw this for a grade, it's really important to pair up these colors in the right order that you're doing them to kind of show that you understand that where they are sort of matters. Remember that this is really important and really cool that right now over here you can see that there's a long red, long black, short red, short black, and medium red, medium black. Which takes us to our second to last part of the cell cycle and our last part of metaphase. And our last part of mitosis. The last part of mitosis is telophase. And our nuclear membranes will start to reform. The cell will start to pull apart from each other. And what is this called? All right, good job, Frank. Yes, it is called cytokinesis. All right, so we've got a long black one in each of them, a short black one in each of them, a medium black one in each of them, short red in each, medium red in each, and a long red in each. We've got our centrioles over here. This is called telophase. Mitosis is now over. We have two fully formed nuclei, and mitosis is just the process of separating the nucleus. Now what kicks in is cytokinesis, which will separate the cells into two daughter cells. Chromosomes will start to unwind, so we really shouldn't be able to see those chromosomes anymore. But for the purposes of us and grading and things like that, I want to know that you know what's in each of these cells. So you should have, since our N was 3, you should have 3 unique chromosomes, but because each of these is 2N, you should have 2 of each of them. All right. This is cytokinesis happening. Remember in plant cells, they just build a brand new cell wall right down the middle there to separate from one another. Okay? Good. So we've got our drawings there. Now let's talk about something else. So what I'm going to do is I'm going to add this picture in here. And this is a picture that we already took a look at. Here's our picture right here. Move that up here. These are actual pictures from like onion cells. Onion cells near the root are really, really, really good and easy to see all of the chromosomes on. And because there's a lot of them doing mitosis right there because it's growing. And when you're growing, you need more cells. And so because you need more cells, your cells are going through mitosis a whole lot. Okay. So that's why onion roots are really, really good to see a lot of cells in mitosis. Whitefish eggs are also really clear and easy to dye and see mitosis in, because the whitefish eggs, in these eggs, you have whitefish growing. And when you're growing, you got to make new cells. And when you got to make new cells, you should see a lot of cells in. That's right, mitosis. Okay. So take a look at this picture here. I'm going to zoom in a little bit. All right. So this is a bunch of cells, all from the root of an onion. This is where the onion is growing from. So we should see a bunch of cells in mitosis, and we can. Let's see here. Let's make a little chart for ourselves. We have interphase, prophase, metaphase, anaphase, telophase. So over here, the key difference between interphase and prophase is that in interphase, you can't really see like the stringy chromosomes, but here you can. Metaphase is easy to see because they're all lined up in the middle. Anaphase is easy to see because they're separated from each other. And then telophase, you should be able to see like a little bit of a cell wall forming in between. But the key is that you can't really see the stringy chromosomes in the nucleus anymore. It becomes a little bit blurry. So let's go over here and let's take a look at this picture right here and see if we can spy how many of our cells are in the different parts of the cell cycle here. All right. Okay, this here looks like an anaphase because they're separated from one another. This looks like a prophase because we can see the chromosomes, but they're kind of still in that nucleus shape. This one right here, it's hard to tell. We may be seeing two cells stacked one on top of one another. So you know what? We're not going to count this one. Aha, this is definitely an interphase. Definitely a prophase. See them here lined up along the middle? That's a metaphase. Prophase, interphase, interphase. Ooh, this is a pretty good anaphase right here. Prophase. This is an interphase. But you know, I'm not going to count these cells because they're kind of ghosty. Interphase. This looks like a telophase has just happened. That cell wall is not very clear in between. Anaphase, prophase, interphase, 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 metaphase, inter, anaphase, inter. This is right at the start of anaphase. They're not very apart from one another yet. Inter. This is ghosty. Pro, ana, inter. This one's neat right here because you can kind of see a little bit of something forming right here. And you start to lose sight of the stringy chromosomes. So I would say this is a really good telophase. Inter. This one's really neat too. I think this is also a telophase because that line in between isn't very clear yet. And they're pretty close to one another, these nuclei. So I would say this is another telophase. Probably another telophase here. Prophase, inter. Here's a metaphase. They're lined up in the middle. Here's a prophase, kind of ghosty. Inter. Anaphase. I could see why you might even say telophase for that one. Inter. Okay. So I'm going to take a minute and add these up. You don't have to listen to me counting them up, so I'm going to stop the video and do that. Why don't you go ahead and pause it and count them all up too? All right. This is what I got. Hey, you know what? If we're off by one or so from one another, we were just doing some quick counting, and that's okay. All right, so let's remember that all of these right here are part of mitosis. So how many total cells do we have here? We've got 10, 19, 23, 61 total cells. So our total number of cells is 61. So what we can do now is we can create this thing called a mitotic index. And in a mitotic index, we take the number of cells in mitosis, which is 23, and we divide it by the total number of cells. So 23 divided by 61. That's going to give us a number between 0 and 1. And so these mitotic index numbers are between 0 and 1. 0 meaning 0 cells are in mitosis, and 1 meaning that all of the cells are in mitosis. And if you remember back to our picture of the cell cycle, you'll remember that most of the life of a cell is not spent in mitosis. And that's why we see a huge number of cells in interphase. Even in a part of the cell where a lot of cells should be going through mitosis, there still aren't a ton of them. All right, so I find this number is 0.377. So even in cells that don't have that should have a lot of mitosis going on, that number still is not super high. So the number of cells in mitosis can tell us a lot. Where should we see a lot of cells in mitosis? Well, think back to yesterday's notes. Well, the last set of notes, which was that we should see cells that are in a repair state in mitosis. We should see cells from a part of our body where you're developing should be in mitosis. We should see cells, oh my gosh, that are in really growing parts of the body in mitosis. But there shouldn't be a ton of them. And so this number will give us an idea of the ratio of cells that are in mitosis. And this shouldn't be very big for a whole bunch of places. Okay. Let's go back to our cell cycle now. Let's go back to our cell cycle. We're going to draw ourselves a quick one down here. All right, so our cell cycle should be most of it in G1. Then we have an S phase, a G2, a mitosis with this little cytokinesis part of it. So our cell cycle, your cells aren't always doing this. Your cells will get locked into different parts of this at different times. And right about here in G1, what we have is what's called a checkpoint. A checkpoint. And over a little bit more. At this checkpoint right here, the cell checks for a whole bunch of stuff. This is called the G1 checkpoint. Are there enough nutrients for the cell to continue? Is the cell being told to grow by different external things, different molecules or hormones? Is the DNA damaged? The cell is always checking to make sure that DNA is damaged. And it's possible for the cell at this point to be told, you're not going to grow, at which point it kind of enters this phase called G0, which is where the cell just sort of lives and rests, and it's just sort of doing its life, but it's not going to come back into this cell cycle. So this is called quiescence, but we don't really need to know that. All right. But if the cell is not told that it doesn't need to grow, then it will continue on. Now, right before it goes into mitosis, it does another checkpoint. This is the G2 checkpoint. And your cells detect a whole bunch of signals from its surrounding environments to figure out if they need to reproduce. If there's too many cells around it, it'll actually detect that pressure and not divide. That's like a checkpoint, against cancer, which is really, really, really important. So is the cell big enough? Is the DNA okay? Those are the kinds of things that it is looking for. And if that's true, if all of that is true, then the cell will continue into mitosis. There's one last checkpoint, though, and it's right here at metaphase. And at the metaphase checkpoint, that has a K in it. At the metaphase checkpoint, it checks to make sure that all of the spindles are attached to the chromosomes in the right places. Otherwise, you end up with cells with the wrong number of chromosomes, which is really, really, really bad. And at that point, if everything's good, goes through the rest of mitosis, cytokinesis, we have 2 daughter cells, and we are good to go. So how does it do this? It does all of these checkpoints using a kind of, that should be an O, using a kind of protein called a cyclin. That's nice and easy to remember. There's a protein called a cyclin that does some of these things. And so what happens is a cyclin activates another protein called a cyclin-dependent kinase. There should be a plus sign here. And when these two work together, they activate another protein that causes the cell cycle to continue. And you might remember that way back when we learned about respiration, ATP uses phosphates, and when it attaches phosphates from itself to another thing, it activates and energizes that particular protein, and that's what this does. This cyclin and CDK together will add phosphates onto other proteins, which causes those proteins to become active. And so if we were to draw a graph of this, let's make this little graph here really quick. We've got our interphase, which goes to mitosis. Looks a lot like this. As the cyclin concentration increases, mitosis is more likely to happen, but after mitosis drops, the cyclin also drops. And so as cyclin activity increases, we get all the way to mitosis. And when cyclin drops off, then the mitosis kind of stops. So there's different cyclins that activate different parts of the cell cycle. There's not just one cyclin, but there's multiple kinds of cyclins that happen. So let's talk about what happens when these kinds of things mess up. When the whole cell cycle makes mistakes and we get problems happening, we end up with cancer. And cancer involves the development of tumors. We don't want tumors, but tumors are these abnormal cell growths. And these abnormal cell growths are the result of lots and lots and lots of mitosis happening because we're getting this uncontrolled cell division. Due to uncontrolled cell division. And these tumors block off how your organs are able to work because they grow. They block tubes, they block blood flow, they block ducts. They stop parts of your body from working because they're getting all crowded out. And as a result of these parts of the body stopping working, they can't do their job, and all of these problems continue throughout your body. Remember that there are these things called mutagens that can cause the DNA of a molecule to change, which could lead to cancer. And these are things like radiation from the sun or from another natural source on Earth, uranium or plutonium and things like that. There are chemicals like arsenic and asbestos and things like that can cause it. But remember that there's also viruses and bacteria, some viruses and bacteria that can actually change your DNA. So now, how is your DNA connected to this? Well, remember that proteins, the instructions to make proteins are found on your DNA. And what is one of the proteins that we just learned about? One of those proteins is cyclin right up here, and cyclin-dependent kinase, or CDK, which is an enzyme. These are both proteins, and if these genes get mutated and these proteins don't develop right, is it possible that the cell cycle does, well, too much? Is that possible? And the answer is yeah, that is. And so the problem is that some of these genes become inactive, and as a result of becoming inactive, the cell cycle is allowed to just continue unabated. And it keeps going and 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 going until you get this giant growth called a tumor. I think it goes without saying that there's some things that we most definitely know of that cause cancer, like cigarette smoke. Remember that when we find relationships for things, we're looking for correlations. So here's a graph, and there's a very clear connection between cigarettes per day and the incidences of cancer. All right. And that graph looks a lot like this. This is called a positive correlation, which means that as one thing goes up, the other thing does as well. I've seen a lot of test answers coming back. When asked about correlations, people say that there is a relationship, but they don't say what that relationship is. This is a positive correlation, which means that the number, as the number of cigarettes per day goes up, so does the incidences of cancer. A negative correlation would be if the line went down. If the number of cigarettes went up, cancer went down, but that is definitely not what it is. Remember that correlation doesn't prove causation. This means that the only thing that we can say on this graph is that there is a correlation between the two, but then it opens up the question, okay, well then, do cigarettes cause cancer? And a lot of research has been done to that, and the answer is unequivocally yes. Now, so how do doctors see cancer cells? Women yearly get this thing called a pap smear, where the doctors go in and take a little sample of cells from their cervix and take a look at it under a microscope, because cancer cells look a very specific way. Okay, let's talk about a couple characteristics of cancer cells, and this will close out the notes portion of this here. All right, cancer cells. So cancer cells, when you look at a cancer cell, has this really big, weird-looking nucleus, because a lot of cancer cells actually develop like way too many copies of DNA than they're supposed to. And so they get these huge nucleus with like a little bit of cytoplasm around it, like not a lot. And so that's one characteristic of them. Normally in your different tissues, your cells are arranged in these nice organized little like layers and packages. But cancer cells, when you look at a bunch of them, like right next to each other under a microscope, end up being all kinds of weird shapes instead of like a nice organized little grouping. So they have a lot of variety in size and shape, and they're really disorganized. Let's see here. Well, normally cells look a very, very, very specific way. I mean, think about how specialized neurons looked, Cancer cells lose that kind of special features, and they develop all kinds of weirdnesses. If a cell is supposed to be nice and round like this, maybe it develops these like arms and things like that. So the cells like lose their special features. And then finally, there's just a lot of them. So where you're supposed to have a certain number of cells, you end up with a ton of cells. and it's not very evenly spread out. I mean, your body is full of lots of borders. Think about it. Your stomach is a very specific shape, and your heart is a very specific shape. And your skin goes down to a specific layer, at which point it's dermis, and under that it's hypodermis, and so on and so forth. And you lose those boundaries with cancer cells. Okay, that's it for today.